For decades, diabetes has been seen as a problem of insulin either not enough or cells ignoring it. This view has shaped every aspect of treatment, focusing on blood sugar and insulin sensitivity. But what if that's only part of the story? New research suggests the real issue may start deeper inside our cells' energy factories. Imagine fixing a car's fuel gauge when the engine is broken. The real problem goes unnoticed. Scientists now believe the root cause may be a failure in our cells' power plants, not just insulin resistance. This discovery could redefine diabetes, shifting the focus from insulin to a crisis of cellular energy. The question is, what happens when our cells can't make enough energy? Meet mitochondria, the tiny power plants in every cell. Their job: turn food into ATP, the energy that keeps us alive. In the pancreas, beta cells rely on mitochondria to make and release insulin. When mitochondria fail, beta cells can't sense sugar or produce insulin. This mitochondrial dysfunction may be the true starting point of type 2 diabetes. Recent studies show broken mitochondria don't just run out of energy, they send distress signals that shut down insulin production. The result: blood sugar rises and the classic symptoms of diabetes appear. This new science suggests we've been treating the symptom, not the cause. Fixing mitochondria could change diabetes care forever. The real culprit may be a cellular power failure, not just insulin gone wrong. Mitochondria aren't just batteries. They're complex, dynamic, and essential for life. Their folded inner membranes create massive energy output from food and oxygen. Think of them as the city's power stations. Without them, everything shuts down. They also control cell communication and even have their own DNA, a relic of their ancient origins. When mitochondria falter, energy drops and harmful byproducts rise, damaging cells further. This creates a vicious cycle of decline: less energy, more damage. In diabetes, this meltdown may be the first domino to fall. Understanding mitochondria is key to understanding and treating diabetes. If you're enjoying this video, don't forget to like, share it, subscribe, and click the notification bell so you do not miss any of our future videos. Thank you. Deep in the pancreas, beta cells act as insulin factories powered by dense networks of mitochondria. After a meal, mitochondria ramp up, producing the ATP needed for insulin release. This surge is like a factory doubling output. Without enough power, production stalls. If mitochondria fail, even a small drop in ATP cripples the cell's ability to sense sugar and release insulin. This isn't insulin resistance, it's a failure at the source. The insulin factory loses power, and blood sugar control breaks down. This insight shifts our focus from insulin's effects to the health of the cell's power grid. Protecting mitochondria could be the key to preventing diabetes. The real problem may be a power outage, not a broken lock. Mitochondria don't just power beta cells, they're the sensors too. When glucose enters mitochondria, turn it into ATP, raising the cell's energy. This triggers a chain reaction: channels close, calcium rushes in, and insulin is released. If mitochondria are damaged, this signal fails, insulin stays locked inside. The beta cell becomes blind to high blood sugar. This breakdown at the very first step leads to persistent high glucose, the hallmark of diabetes. The new research shifts focus from insulin's effects to the cell's energy-sensing machinery. Fixing the sensors may be the breakthrough we need. Scientists long suspected mitochondria were involved in diabetes, but the link was unclear. A landmark study changed that. Researchers induced mitochondrial failure in healthy beta cells, and diabetes followed. The result: beta cells lost their identity and stopped making insulin. This proves mitochondrial failure isn't just a consequence, it's a cause. The story of diabetes now starts deep inside the cell, not at the cell surface. This insight opens new doors for therapy. Protecting or restoring mitochondria could prevent or even reverse diabetes. The future of diabetes care may lie in fixing our cells' power plants. The Michigan study produced compelling visual evidence using advanced electron microscopy. In healthy cells, mitochondria appeared well-defined with densely packed internal folds. In diabetic subjects, the mitochondria were almost unrecognizable, small, swollen, and fragmented. This physical deformation was visible proof of their functional decline. A mitochondrion with fewer cristae is like a factory that has had most of its assembly lines ripped out. Similar damage was observed in the liver and fat tissue, key players in metabolic health. This explains why type 2 diabetes is a systemic disease, contributing to insulin resistance and weight gain. The Michigan study revealed that damaged mitochondria send distress signals to the cell's nucleus. These signals hijack the cell's programming, triggering an unexpected response. Instead of dying, beta cells de-differentiate, losing their specialized characteristics. They shed their identity as insulin factories, conserving resources in a desperate survival strategy. Researchers termed this phenomenon cellular amnesia. The cells forget their purpose, their genetic instructions silenced. This explains why beta cell function declines, even when cells are present. Cellular amnesia suggests that loss of insulin production might not be irreversible. Blocking distress signals could reawaken dormant cells, restoring the pancreas's ability to produce insulin. To prove mitochondrial dysfunction caused cellular amnesia, the University of Michigan scientists designed a comprehensive experiment. They devised a three-pronged strategy to sabotage mitochondria in healthy lab mice. Each method targeted a different aspect of mitochondrial health. The first targeted the mitochondria's genetic material. The second disrupted the cell's quality control system. The third interfered with proteins maintaining mitochondrial structure. All three methods led to the same outcome. Beta cells de-differentiated and stopped producing insulin. This consistency proved that mitochondrial health is essential for beta cell function. The energy crisis was the trigger. The Michigan study targeted the mitochondrian's unique DNA. Mitochondria possess their own DNA, containing the genetic code for 13 essential proteins required for energy production. Researchers disrupted a key gene within this mitochondrial DNA in mice beta cells. This precision sabotage halted cellular respiration. The mitochondria could no longer build essential components, causing the electron transport chain to fail and ATP production to plummet. Crippled mitochondria sent stress signals, alerting the nucleus. Beta cells began to change, switching off genes responsible for insulin production and secretion. The cells were de-differentiating, losing their function to survive the energy famine. This linked mitochondrial genetic integrity to cellular identity, proving the damage initiated within the mitochondrion. Mitochondrial blueprint flaws triggered cellular amnesia, inducing a diabetic state. The link was direct and causal. The second method targeted mitophagy. This is the cell's quality control and recycling program for its mitochondria. Healthy cells monitor their mitochondrial population, identifying any that are old or damaged. These faulty powerhouses are then broken down, a process known as mitophagy. The researchers used a genetic technique to disable this cleanup crew, blocking the mitophagy pathway. Dysfunctional powerhouses accumulated, producing damaging free radicals. The outcome was identical to the first experiment. The buildup of damaged mitochondria triggered the same integrated stress response. The beta cells began to shed their mature identity, silencing the genes for insulin production. This demonstrated that the trigger for cellular amnesia wasn't just a lack of energy, but also mitochondrial stress itself. The third prong involved disabling key proteins responsible for mitochondrial quality control. Despite the different causes, the result was eerily familiar. Fragmented mitochondria, falling ATP levels, and beta cells shutting down insulin production. The consistency across all three methods revealed a profound biological principle: a master switch, a unified stress response pathway. The cell interprets mitochondrial issues as an existential energy crisis. The response is always the same: a retreat to a less energy-intensive state. This pathway, the integrated stress response, ISR, is an ancient survival mechanism. The study provided evidence that the ISR links mitochondrial failure to the loss of beta cell identity in diabetes. This discovery is hopeful because a single pathway responsible for cellular amnesia presents a clear, druggable target. What if we could intercept the message that tells the beta cell to shut down and reverse the cellular amnesia? This question formed the basis for the search for a drug that could do exactly that. Researchers at the University of Michigan moved to the most thrilling phase of their work after identifying the integrated stress response, or ISR, as the master switch that triggers cellular amnesia. The search was on for a compound that could specifically interrupt the ISR signaling cascade, acting as an antidote to the mitochondrial distress signals. The compound is known as ISRIB, which stands for Integrated Stress Response Inhibitor. ISRIB was designed to block the downstream effects of the ISR pathway. It effectively puts noise-cancelling headphones on the nucleus, preventing it from hearing the constant alarm bells and overreacting by shutting everything down. ISRIB works by releasing this brake, allowing the cell to continue producing the proteins essential for its mature identity and function, even in the presence of ongoing stress. They began treating diabetic mice with ISRIB. The hypothesis was that ISRIB could reverse the course of diabetes. The results of the ISRIB treatment exceeded even the most optimistic expectations. The researchers administered the drug to the diabetic mice, and within weeks they began to see a dramatic transformation. The blood sugar levels started to decline steadily, returning towards the normal, healthy range. When scientists examined the pancreatic beta cells, they found the cells were waking up. The genes responsible for insulin production, which had been silenced, were now active again. When challenged with a glucose test, their newly awakened beta cells responded beautifully, releasing the appropriate amount of insulin. In effect, the ISRIB treatment had reversed diabetes by restoring the underlying biological function that had been lost. This is a landmark discovery because it represents a completely new therapeutic strategy. This offers a radical new hope for treating type 2 diabetes in humans. The implications of this discovery are truly vast. It suggests a future where type 2 diabetes might not be a chronic, progressive disease, but actually a reversible condition. A treatment based on ISRIB could potentially restore a person's own natural ability to produce insulin. This would be a paradigm shift, moving away from managing symptoms and towards a therapy that actually repairs the underlying cellular defect. It could also address the systemic nature of the disease, as ISRIB may help restore mitochondrial function in liver and fat cells, tackling insulin resistance and weight gain simultaneously. This breakthrough opens the door to developing a new class of drugs that target the root cause of metabolic disease. The success in mice provides a powerful proof of concept, validating the idea that protecting cells from mitochondrial stress is viable and highly effective. It gives researchers a clear path forward, focusing on drugs that can block the integrated stress response in humans. This could fundamentally change the lives of millions of people who are living with diabetes and its complications. However, it is absolutely crucial to temper this excitement with a strong dose of reality and caution. ISRIB itself is currently an experimental drug. It is not approved for human use, and its long-term safety profile is completely unknown. While the results in mice are incredibly promising, what works in laboratory animals doesn't always translate to human biology. The journey from breakthrough to pharmacy shelf is long, complex, and expensive, often taking many years of rigorous clinical trials. Therefore, while we celebrate this scientific advance, we must be patient. Human clinical trials are the essential next step to determine if this approach is safe and effective. Scientists will need to evaluate potential side effects and determine the correct dosage. The road ahead is still long, but the Michigan study has provided a clear map. For the first time, we have a tangible strategy for a genuine cure, offering a beacon of hope where before there was only management. The Michigan study's implications stretch beyond diabetes. Mitochondrial failure can trigger a cellular identity crisis, providing a new lens for viewing chronic diseases. The brain, an energy-hungry organ, is sensitive to mitochondrial dysfunction. Failing mitochondria are linked to neurodegenerative conditions like Alzheimer's and Parkinson's. Mitochondrial dysfunction in fat and liver cells contributes to type 2 diabetes and metabolic syndrome. Mitochondrial dysfunction is a potential common denominator underlying major chronic diseases and aging. By rejuvenating these cellular powerhouses, we may revolutionize the treatment of seemingly unrelated diseases. With such profound implications, the direction of future research is now clear and galvanized. The absolute priority is to translate these incredible findings from laboratory mice into tangible therapies for human patients. The immediate next step involves moving beyond animal models and beginning preclinical work using human cells. Researchers are now taking cells from patients with type 2 diabetes and growing them in the lab. They will then treat these cells with ISRIB-like compounds to see if they can replicate the reawakening effect observed in mice, coaxing human beta cells out of their state of cellular amnesia. This is a critical validation step before human trials can begin. Simultaneously, the race is on to develop and refine drugs like ISRIB, specifically for human use in metabolic disorders. While ISRIB itself is a remarkable research tool, it may not be the ideal candidate for a long-term human medication. Pharmaceutical scientists are now working to create new molecules that are more potent, have fewer potential side effects, and can be targeted more specifically to the affected tissues. The goal is to design a second-generation drug that retains the benefits while ensuring maximum safety for chronic use. This research also opens up exciting new avenues for diagnostics. If mitochondrial decline is the earliest step in diabetes development, then detecting this decline could provide a new way to identify at-risk individuals long before their blood sugar begins to rise. Scientists are exploring novel techniques to measure the health of a person's mitochondrial network. Such a test could act as an early warning system, allowing for preventative interventions before irreversible damage occurs. The ultimate vision, as articulated by experts like Dr. Solimanpour, is nothing less than a complete rewriting of the textbook on metabolic disease. The focus is shifting from managing symptoms to restoring health at the most fundamental cellular level. The journey from the lab to the clinic is long and filled with challenges, but the path is now illuminated. The next decade of research promises to be one of the most exciting in the history of medicine, as we learn to harness this knowledge to potentially reverse diabetes and enhance human health and longevity. The prospect of a drug like ISRIB that could reverse diabetes by reawakening dormant cells is undeniably thrilling, offering a profound sense of hope for the future of medicine. However, the journey to a clinically approved treatment is long, and we cannot simply stand by and wait. The wonderful news is that the very science that illuminates this future path also provides us with a clear, actionable blueprint for what we can do right now to protect and enhance our mitochondrial health. One of the most potent ways to boost your mitochondria is through exercise, particularly high-intensity interval training or HIIT. Nutrition also plays a pivotal role. While a balanced diet is key, certain nutrients and supplements have been shown to directly support mitochondrial function. Another powerful strategy is intermittent fasting or time-restricted eating. It's a natural reset button for your cellular health, a way to declutter your internal machinery and ensure it runs with maximum efficiency. These lifestyle hacks are our immediate tools in this fight. We are standing at the dawn of a new era in our understanding of health and disease. The story of mitochondria and diabetes is a call to action. Our daily choices, the food we eat, the way we move, the times we rest, have a profound impact on our lives. This is about empowerment, recognizing the incredible influence we have over our own biological destiny. This research should inspire us to become more proactive in our own health journeys. Has diabetes affected you or your family? This new science offers a present-day strategy for resilience. I urge you to share this information. Discuss this with your doctor, share it with your family, and become an advocate for a more foundational approach to wellness. You are the custodian of trillions of tiny power plants, and you have the ability to help them thrive. The power to improve your metabolic health is already in your hands. Become the architect of your own cellular vitality. If you enjoyed this video, don't forget to like, share it, subscribe, and click the notification bell so you do not miss any of our future videos. Thank you.